get started. Slide, please. The goals of the series, this has not changed since we began, which seems like a long time ago now. We're going to provide scientific and patient-oriented perspective of myeloma features, and its biology, set the stage for other presentations in this series. Useful information about newer treatments and availability. Dr. Donna Reese is the medical director of CMRG, and she has her finger on everything that's coming down the pipe. We want to provide a framework for discussion about immunotherapies and how our patients can access them via clinical trials and access programs. And as always, most importantly, provide a forum for all of you who are stakeholders to ask any questions. Slide, please. So today, Dr. Reese primarily will be talking about our two basic types of immunotherapy that we see in treating multiple myeloma. The first would be what is referred to MOABs, which is monoclonal antibodies. This is an antibody molecule that's made in the laboratory, and it actually can recognize the myeloma cell. The patient's often given a loading dose, either intravenously or subcutaneously, which means under the skin, followed by less frequent doses every 2 to 4 weeks. This is given repeatedly over a relatively long period of time. So MOABs, as Dr. Reese will discuss further, can be plain or naked. They attach to a toxin like belamaf, and they also can be bispecific. The other thing that we'll discuss a bit tonight, and there'll be further discussion in other seminars, is regarding T-cell therapy. A few years ago, CAR-T was something that many myeloma patients and family members were waiting for. CAR-T is the most developed type of cellular immunotherapy that we have so far. Immune cells, which are the patient's T lymphocytes, are collected from a person and then reprogrammed in a lab to recognize and kill myeloma cells. So this is incredible science, all because of all the clinical trials and work that's gone on in the labs. And then the CAR-T cells are given back intravenously, and then with the idea that then they go and directly kill the myeloma cells. This usually, CAR-T is only given one time as a treatment. So for those of you that have had autologous stem cell transplants, not the same scientifically, but the fact that you get your cells back, but this time these cells are now reprogrammed in a lab. Slide, please. Dr. Reese will now talk more about how the MOABs target the surface on the myeloma cells.
Yeah, and I feel like we've been leading up to this hot topic ever since we started this session, and I appreciate everyone's patience because this is right at our fingertips for us to start being given to those that need it. But in a little bit of defense, we wanted to set the stage for common understanding of myeloma, how we monitor it, et cetera, et cetera. But to get into the field of immunotherapy, my first couple of slides just summarize a few points we talked about last time for those that couldn't join. So we talked about monoclonal antibodies. Those are those Y-shaped molecules. And the first ones we had were plain or so-called naked antibodies. They're not attached to anything. They just stick on myeloma cells. And the first target was CD38. That's one of the markers sticking off the myeloma cell, of which there are many. We want to pick a marker that's only on myeloma cells and not on critical organs, or the treatment we would give, the antibody could recognize heart and kidneys and cause havoc. We just want to find a marker that's on the cancer cell. Now, these naked antibodies can kill the myeloma cell to some extent, but they almost serve as a flag to call in the rest of the immune system to do a lot of the killing. Nevertheless, they're quite effective. And we now have in various settings daratumumab, and the brand name is Darzalex, or isatuximab, or Sarclisa, which we are delighted to have, particularly when they're combined with our older drugs. And by the way, they're going to be combined with the BiTEs. They have really enhanced the killing power of our combinations, be it mixed with lenalidomide Revlimid, mixed with bortezomib or Velcade, with pomalidomide, with carfilzomib, on and on. You can combine them with anything and you get better results. Next slide, please. We also talked last time that the second kind of antibody that Suzanne talked about after naked, and that's actually a term that's used in the literature. Usually terminology in myeloma isn't quite as exciting, but anyway. The other kind is what we call an antibody drug conjugate, where an antibody has a toxin stuck on the end of it. And here I've shown a cherry bomb. We talked about this quite a bit last time. And the target for these antibody, or this antibody, has been another marker on myeloma cells called BCMA. And so this is more powerful than a naked antibody. And so the ADC, in this case, belantamab, and the other names are there, attaches to the surface marker and then adds a poison to the cell. And these are more potent. And we talked last time that the toxin, or the cherry bomb, is the one that irritates the eyes. Next slide. So finally, we're going to talk about BiTEs. That is an abbreviation for bispecific T-cell engagers. Now, what that means is, and I don't have a pointer here, but if you look at the Y-shaped antibody that I think you'll recognize, usually each of the ends that stick on something stick on the same thing. You know, the CD38 or the BCMA, but a bispecific has been engineered that one arm sticks on the T lymphocyte. And that's a cytotoxic cell. It's a normal cell that can kill invaders itself. And the other arm sticks on the myeloma target. And the first BiTEs we have stick on BCMA. And they bring these two different cells together, and the T cells don't like a cancer cell. And so they release little chemicals that are called cytokines, messengers, that help kill directly the myeloma cell. So it's a direct reaction. Some of those, if you're interested in the names, are called perforin or granzymes, and they're how T cells kill, you know, other invaders, viruses and things like that. But we're leveraging this to kill the cancer cells. This is really a brilliant technology. And the BiTEs have now been made to several targets that we'll talk about, and the race is on for pharma. To every pharma company wants a BiTE against myeloma. My only hope is that will bring the cost down if there are all these competitors, but that remains to be seen. Also keep in mind that these little chemical mediators that are released, I kind of describe it sometimes as they're creating killing of the myeloma cell, but they're also causing smoke and fire. And it's not necessarily the ones named here, but there are other ones I've not shown that can cause the fever and other reactions that are the unusual side effects of bispecifics. Next slide, please. So again, the first target studied was BCMA, and many of the BiTEs, I'll give you a list later, that have been made that all target BCMA. Now, these antibodies have slight differences, but they have the same target. and I'm getting ahead of myself, there's a lot of interest in, if one BiTE doesn't work against BCMA, will another brand work? That's all for the future, but they're not identical, but they all attack the same marker on the myeloma cell and bring the T cell there, right close by to do the killing. Next slide, please. So where do these BiTEs fit in? And as we've talked before, new treatments for cancer are inevitably studied and approved when other treatments aren't working. It's felt that's the lowest risk. I mean, if you have good treatments, you don't want to disturb those with a new drug that you don't know how well it will work. You don't know if the side effects are going to be okay. So in Canada, Bispecifics, the two that we have, that we'll talk about their names a bit more in a minute, are approved for fourth line treatment. And I've just shown the basic outline. We've gone pretty much through all these with our previous 6 sessions. So hopefully what we've used before in their outcomes has been discussed at some point. We are a little bit lucky in that in the US, these are approved after 4 lines of treatment for 5th line of treatment. So how we got it 1 line shorter, I don't know, but we'll take it. Next slide, please. So what are the 2 BiTEs that are approved by Health Canada? The first is made by Janssen. Its full name is teclistamab. We all call it TEC, and I'm going to call it TEC for the rest of this session. The second one is made by Pfizer, and it's called elranatamab, and we call it ELRA. So the indication for Health Canada is for people with myeloma that's relapsed, that have received at least 3 prior lines of therapy, and at some point have received a proteasome inhibitor, that's Velcade, carfilzomib, or ixazomib, an IMiD, usually Revlimid or lenalidomide, but that can be pomalidomide or technically thalidomide, although we don't use that much, and an anti-CD38 antibody, so either daratumumab or isatuximab, and their most recent treatment, whatever it is, isn't working and the myeloma is growing. Right now, these 2 BiTEs are under review by CADTH, which is the health technology assessment body that reviews all the data and the cost effectiveness and recommends to the provinces whether it should be paid for, and it now has to follow the Health Canada wording. Then each province in turn decides whether they agree with that, and if and when, and they can wiggle room it a bit. And some of you may have responded for the patient feedback. We have pushed very hard with our CMRG feedback that we want people to have access to, if they've had belamaf, which is BCMA, that they can get another BCMA drug like a BiTE. Don't automatically cut people off, which is always the dynamic, because since it's not on the market yet here, we don't have the list price that will be in the pharmacies. Now, what actually is negotiated is not what's on the list, but I promised Suzanne I wouldn't talk all about the US, but in the US, these drugs are $40,000 a month. And they are currently approved to be given continuously. So 40,000 times 12 months a year, they're basically priced like CAR T cells. So we can see the challenges in trying to get all these drugs to our patients that need them with this very, very high price, which there will be compromises made, of course, but you know, we want our patients to have access to as many of these as they need, but realizing each one of these types of treatments is very innovative, but very expensive. Next slide, please. Now, how well do they work? Well, I've summarized the two key trials. And I'm sorry, this is med speak, but I didn't know how else to present it. But tech was studied first, and all these trials have nice names. This was the Majestic one trial, and it was reported in 2022. And just to give you an idea of the patients treated, and keeping in mind, we're going to use this earlier, and generally drugs work better the less treatment someone's had. The myeloma cells just haven't become as resistant to everything. So the median age was 64, and I think what's notable is patients up to age 84 were treated. If you look at the proportion who's over 75 years of age, it's 14%. These individuals had had a median of 5 lines of treatment, ranging from just 2 to 14. Overall response rate, over 50%, 53%. And the median benefit, that means half people longer, half people shorter, was about 1 year. And if people were in the 50% that responded, of course, it tends to be longer. So elranatamab, the other approved bispecific, the results were reported in 2023. This is the Magnetism 3 trial. Here, the median age was a titch older, and almost 20% of people were over 75. Again, median 5 lines of treatment, up to 22 lines of treatment. Overall response rate, very similar, 61%. Median benefit, very similar. 15 months, it's not quite the median been reached, but it's going to be a little longer than 15 months, it looks like. But again, if people were in that 60% who responded, it tends to work longer. If you stand back and I didn't put this in the slides, and look at all the bispecifics that have been studied in the first wave of studies, in the people with a number of lines of treatment, 5, 6, 7 lines is where it's been studied, the response rate is pretty consistent, around 50 to 60%. So right now we can't say, oh, someone should have this BiTE, someone should have that BiTE. It's pretty consistent in the response rate. Next slide, please. So, Suzanne, you have to tell me when my time's up.
I will. You have this slide and one more.
Okay, thank you. So what's the downside of BiTEs? There are unique side effects because this is something brand new for the body. I just wanted to try to provide a high level overview of these two, because you'll hear that discussed a lot. And if any of you have received these drugs on a trial or now access programs, you'll hear this. So cytokine release syndrome happens because those little mediators that are released from the T cell and call in other forces lead to a very hyperactive immune system for a while. And this occurs quite early after the bispecifics given. And that's why we start out with a little dose, and then a certain days later give a slightly bigger dose, and then it differs with the bispecific, but we have what are called step-up doses because you don't want to give the full dose at the beginning when there's a lot of myeloma cells around. You'll get more smoke and fire. So this has been worked out quite nicely from a number of years ago. And there are standard pre-meds that help suppress the smoke and fire. The three main symptoms are fever over 38, low blood pressure, and low oxygen level. And I don't want you to spend a lot of time on the picture of the person's body there, but there is a grading system that if you just have fever, that's grade 1. Grade 2, if you have a little bit of low blood pressure that you just give some IV fluids for, or a little bit of low oxygen where you give a little bit of nasal prongs, that's grade 1 or 2. And then it goes up to, much more serious where people need vasopressors, that's chemicals to keep their blood pressure up, and more oxygen. What is really good about the BiTEs is although CRS is very common, 50 to 70%, it's almost always grade 1, occasionally grade 2. You don't, the rate of grade 3 is very, very low. And it's less severe than with CAR T cells. And I think the best news of all is there's an antidote. There is, think of it as an anti-cytokine drug that's actually used in certain autoimmune diseases like rheumatoid arthritis to cool off the immune system, a drug called tocilizumab that we call Toci. You give a dose of that and the CRS in the majority of people melts away. And it's very gratifying to see if someone, even if they just have fever, and we have moved more and more clinicians with grade 1, even if it's just a fever. Who wants to have a fever for 3 days? You can give a dose of Toci and it goes away. So all of this, I've told you the worst, but with the bispecifics, this has definitely become manageable. It's not scary anymore, although centers are learning how to do it. And as I'll discuss a bit later on, this was the reason patients were always admitted for the first few doses of bispecific, so they could be watched for CRS and what I'll show on the next slide. But as we become more familiar with it, I think we can appreciate these bispecifics are going to be applicable to, I think, the vast majority of people with myeloma at some point. We don't have beds to keep people in 5 to 9 days each time to start a bispecific. We're not going to get those beds. So we have to find a way to give these as an outpatient. Next slide, please. So the other big side effect is what is called immune cell associated neurotoxicity, nerve toxicity. Now, this again is much more common with CAR T, and fortunately, it is less common with myeloma CAR T than lymphoma CAR T. But the cause, it's often with CRS. People may have fever. It's a little bit later than CRS, and people get headaches. They can get confused, not talk as much, not concentrate, feel shaky, rarely in severe cases have seizures. It's treated with steroids. And if there's CRS, people will get Toci too, but Toci is specific for CRS. Now, I don't want to go into the grading, but if you're on these drugs with the step up, you might have participated in these simple screening tools used to make sure, it's hard when you're just talking to someone to know if they're confused sometimes. So very simple questions. These are standardized. Does someone know where they are, the date? Pick out 3 objects that are very obvious and have the person identify them. Follow simple commands like point to your nose. One of the best screenings is to ask people to write one sentence, any sentence, and do that several times a day. And then count backwards, not by sevens like I guess they do for driving while under the influence, but you can count back by tens. So that's good. And this is a part of the scoring, but just to kind of let you know what you could have done just to be sure things are okay. These are good standardized international screening tests, but the good chance is that ICANS won't happen with these bispecifics. Next slide, please. I think that's back to you, Suzanne.
Yeah, thanks, Dr. Reese. I just have a quick question that I have, and I don't know if anybody else sort of thinks the same way. Thinking about both the Majestic and the magnetism trials, and I know that these were patients that were heavily pre-treated with at least 5 lines of therapy, and we know that the myeloma cells get a little bit more fractious and evil as time goes on, and they sometimes become a little harder to get under control. Do you think if patients had been treated earlier and they weren't sort of worn out from having years of treatment, that the results might have been even better than the results that we got?
Well, that's what we're hoping. But that data is just coming out. And remember, this is approved in the US for
Yeah,
after 4 lines of treatment. So
yeah,
those earlier patients, we just don't have enough data. But I think it also, some people think it has to do with how many myeloma cells. I mean,
right.
if someone has had 7 lines of treatment and their disease is really, there's a lot of myeloma cells, there's more to kill and more smoke and fire. These drugs, as I'll show at the end, are being moved up to first line treatment. And I'll be able to address that a bit later, but that's the hope.
Yeah,
When you look at daratumumab
yeah,
by itself in advanced myeloma, the benefit is 4 months of disease control.
yeah,
Now you combine it with everything and you get unprecedented results. So,
absolutely.
I think we're just seeing the beginning, but that obviously is the hope,
Yeah,
that less cancer cells to kill, less smart cancer cells. Back to your point, have they learned to outsmart everything, including immunotherapy? That's the hope. And we are ready to start collecting real world data in our Canadian patients, who will be getting this a little bit sooner, we hope, than patients in other countries. And we'll be
hmm.
able to look at that. We just have to get the data forms finalized and get things rolling.
Well, thank goodness for 2 things. A,
And
we're in
what
Canada, that our patients will be able to receive these drugs hopefully without having to cough up $40,000 a month, and B, that we have CMRG and other researchers looking at all of this. So it's very, very exciting. I'm going to move on a little bit, and I know that myeloma patients and caregivers of loved ones with myeloma are, repeatedly, clinical staff like myself and Dr. Reese, we're always harping on the idea of preventing infection and recognizing infection, and also, responding very, very quickly if there's any signs of infection. And this is very, very true when patients are receiving BiTEs. In fact, it's up to 80% of patients that will be on a BiTE that will develop some type of infection. And we know that an infection that is recognized early and treated early is certainly much better than sort of leaving things. And when I was in my clinical position at Princess Margaret, and I would see patients sometimes twice a week, and they would come in and they'd be very, very ill, and I was always a bit frustrated and saying, why didn't you call? Why didn't you go to the emergency department? And very often I'd hear, well, I knew I was going to see you today, so it was only 2 days, so I thought I could wait. So really, really important to not leave things. We hear often, I didn't want to bother the doctor. I didn't want to sit in the emergency department, which we all understand. None of us want to sit in the emergency department, but it's very, very, very important. Being proactive, trying, really, really working towards not getting infections. Princess Margaret has very, very specific guidelines for prevention of these infections when taking BiTEs. Medications are given prophylactically or in, ahead of the game before people get sick. First, we will have patients on valcyclovir, which is an antiviral medication, and it's given on the first on day of the first step-up dose, and then will continue for a month after cessation. So this will go on at least for 1 month after the treatment, but it may be continued beyond that discretion of the hematologist. So, we have viral infections, and we all lived through our COVID dilemma, and that was a virus. So we want to stay on top of things, and that's taken, they're big blue pills, but certainly easy to swallow, and it's given by mouth twice a day. Then we look at bacterial infections. What do we do? What do we give to prevent bacterial infections? So it can be one of two antibiotics, which are commonly used. The first is ciprofloxacin, and that's also given 500 milligrams by mouth twice a day, or levofloxacin, which is also given at 500 milligrams twice daily. And again, this will start on the day of the first step-up dose and continue for a minimum of 3 months. So through the research, we know that patients will still be at risk to develop a bacterial infection for up to 3 months after the BiTE therapy is discontinued. Pneumocystis, PJP is a specific type of pneumonia. It can be really life-threatening, and patients will start on a Septra double strength. So the DS is for double strength, and it's one tablet, and the tablet will say 80 milligrams over 160 milligrams, and that's given by mouth on Monday, Wednesday, and Friday. There are a number of people that have a sensitivity or allergy to Septra, and if they are not able to take Septra, or they're very cytopenic, meaning their blood counts are very low, they might be able to go on atovaquone, which is a liquid, and it's given just 10 milligrams, which is a third of an ounce daily. Or the other thing, if people can't tolerate Septra, they may be able to receive something called pentamidine, which is an inhaled medication that we certainly give at Princess, or they certainly give at Princess Margaret, and that's given once a month. So again, this begins right at the beginning of treatment and continues to at least 1 month after treatment is finished. But again, all of these are guidelines. The hematologist, the myeloma specialist, may have a different plan for specific patients, depending on their history, depending on how well they are, etc. Another type of infections that patients can get are fungal infections. And a very, very common and not really life-threatening, but very uncomfortable, is thrush. Fluconazole is an antifungal medication, which is given daily at 400 milligrams. Patients that have had a prior history of systemic or extensive localized fungal infections will start on this. But if you have any signs of thrush and you're not on fluconazole, you want to be able to recognize it. Often it will start as thrush in the mouth. And you want to let the team know that you have this sort of white plaquey covering of the tongue, and it's the taste of food might be very certainly not what you expected and can make swallowing uncomfortable. The other thing that is done prophylactically for patients on BiTEs is immunoglobulin replacement. So it can either be given intravenously or be given, and that would be given monthly, or a weekly immunoglobulin under the skin or subcutaneously. The recommendations at Princess Margaret is to routinely start within 1 to 2 weeks of the first step-up dose and continue for a minimum of 6 months after cessation of treatment. There are many patients with myeloma that are already on IV or subcutaneous immunoglobulins. And these are all things that can be discussed with your team. We want to ensure that, first of all, patients have their immunizations. I can't stress enough how important this is. I know during the pandemic, there was many, many, many things on the World Wide Web that made people really, really nervous about immunizations, etc. But for myeloma patients and their caregivers, not just the patient, but their caregivers, because as a caregiver or a clinician, we don't want to get sick and then pass it on to the patient. So we want to look at Prevnar 20, Shingrix vaccines. You always update your COVID-19 vaccinations, your influenza boosters, and if you're over 65 or have, you're immunocompromised, many patients can, certainly if you're over 65, you can get the stronger influenza vaccine. And we want really people to have those vaccinations prior to starting any treatment. So that's something to talk about. If you're in clinic and sort of talking to your team about maybe in the next little while we'll be looking at BiTEs, talk about immunizations and with your physician or your nurse or nurse practitioner, and to make sure that you're always up to date. And everybody, again, out there who's a patient or a family member or a caregiver, we all need to ensure that all of our immunizations are up to date. Also, patients will have screening blood work for prior infections for hepatitis and CMV. And we need to know what patients, if they've had these prior infections, so that they can be managed appropriately. Slide, please. So absence of fever does not exclude infection. So many times people say, well, I didn't get a fever. Sometimes, and this is really for patients that may be sicker or that may have recently been on certain types of treatment, their white count can be very low. And when your white count is low, ironically, the patient may not get a fever. So don't ever think, well, I don't have a fever, so I must be fine. Anything more than a runny nose, anything more than a runny nose, your chest is heavy, you've got, coughing, anything at all, we would much rather see you in either the emergency department or in clinic and get on top of things really quickly. So wherever you're being treated, I'm sure you have phone numbers or instructions of what to do if you're worried about an infection. And often older patients have pneumonia, And I've seen it many times. And I was told, oh, I just sort of felt I've been tired and I haven't felt very energetic, but I just sort of feel bad, and they haven't even coughed. And we send them for a chest CT urgently, and sure enough, they have pneumonia, which needs to be treated urgently. Some of the things that we will do in clinic is we want to check on blood pressure. A patient who has what we call what when a patient stands up and their blood pressure drops significantly, we that alerts us to something that is going on. So your nurse might have had instructions from the doctor or nurse practitioner to do a sitting and standing blood pressure, and that's why we do it. We want to see what the difference is if the patient has something called postural hypotension, meaning that when they stand up, blood pressure drops down, they feel a little wonky. And that can be indicative of an infection. We also will have the patient sort of walk around clinic with the pulse oximeter thing on their finger to see what happens. Does the oxygen saturations, do they drop with any sort of activity? Because the patient might not feel any different, but we can see what's going on. CT of the chest if no localizing signs or symptoms. A plain X-ray, is certainly fine for a relatively well person, who goes into the emergency department and they've had this cough for 3 weeks, but they don't have myeloma. We really, really prefer to do a low dose CT of the chest, because often a plain chest X-ray can miss a very subtle infection, and because if we just send someone for X-rays and we miss that early pneumonia and we don't see the patient again for a couple of days, things could really be getting out of hand. During COVID, we all had nasopharyngeal swabs. Nobody likes them. Nobody likes to do them to patients. We don't like getting them, but it can certainly tell if patients are positive for COVID-19, which is still in the community. It's just not as, thank goodness, as deadly as it was. And there's other viruses that we can determine doing nasopharyngeal swabs. And then you really, really want to remember family members and neighbors and friends. They love you and they want to be around you, but always, always, always be very, very um, firm that if you're sick, we don't want you to visit. And because if I was exposed to a virus, I'd be fine. But if our patients, especially if they're having BiTE therapy, are exposed to the same virus, they may not be fine. And if we have a patient that's unwell and the CT chest doesn't show anything, we're not just going to sort of say, well, everything's fine, nothing's positive. We're going to continue to try and figure out what's going on, what is making this patient feel so unwell. And any patient that's on a BiTE therapy regime really needs close follow-up. So, patients might be asked to come back to clinic even the next day or the day after that, or have a follow-up phone call with the clinic nurse, however your organization or your myeloma treatment area manages things. Slide, please.
So while we're changing the slide, I just wanted to sort of add on, why are we doing this?
Absolutely, thank you.
We typically have preventative antibiotics with some treatments. We always give acyclovir or valacyclovir to prevent shingles and oral herpes with Velcade or carfilzomib, because we know those drugs predispose to reactivation. And people on daratumumab will get something to prevent shingles, an antibiotic like acyclovir, acyclovir, and we'll get Septra to prevent PJP pneumonia. But why are we going all out with this? The reason is these new bispecifics are so potent and so good at killing plasma cells. They kill the cancer cells very well, but they also can't tell the difference between a good plasma cell that makes our good antibodies, of which we still have some, even with myeloma, and the bad ones. So people are losing a part of their normal defenses to infection more than any other treatment. Daratumumab does it a bit. It gets good and bad plasma cells. And we do need to have some antibiotics. We do see unusual infections, but it's a magnitude greater with these bispecifics. And it's the flip side of why they're so good for killing myeloma cells. You might pick up that when you get a vaccine, it makes you stimulates your own good plasma cells to make antibodies against whatever the vaccine's for, shingles, flu. Well, if you're getting a bispecific, you don't have good plasma cells to make the antibodies for your own resistance. So that's why we want the vaccines ahead of time, to give them a little time to take. And also you will find in other countries, they do not give everyone IV immunoglobulin, IVIG, or sub-Q IG, but we have been trying to do that in Canada because people's own antibodies, the good ones, go down with these treatments to quite low levels. So if we can give other people's antibodies, and it's quite safe, you can't transmit diseases like hepatitis or HIV through immunoglobulin. You're giving other people's antibodies to all those good things out there, whatever the donors had, be it measles or some COVID probably, or in bacteria. So we, particularly when we're moving to the outpatient bites, I think we sort of move to give everyone replacement anyway to keep them safe. And there is some data to support that. And we're hoping our CMRG database survey that focuses on infection will support doing that. But that's why we're so diligent. And when we do that, these drugs can be given safely. And we're educating the community, the local emergency rooms, and the community docs, because local emergency rooms are very good at taking care of someone with leukemia with a low white count. They know exactly what to do. They know exactly what cultures to draw, what antibiotics to give. This is a different world. And education, education, education. So all that's in progress. So I just wanted to show you in the next few slides a bit more of how can Canadians get these good drugs. The easiest way, although it's not open door completely, is there are access programs, both for tech and LRA, where pharma has given the drug free of charge, which is fantastic. But usually when drugs are funded, there's money provided to cover all the aspects of care, the pharmacy mixing it up, the nurses to give it in clinic, and other things. Now, the access programs may well give the TOSI, that's the antidote, and it's not cheap either. It's several $1,000s, but that is nothing that's funded anywhere else in the province. It's funded for rheumatoid arthritis, but the cancer centers are, if I may say so, eating much of the cost in order to get the neediest patients these drugs before funding. And we are blessed in Canada that we may get these access programs, because otherwise we do wait till the CADA funding comes through, as we've all seen after Health Canada approval. And this is available in a number of centers. It's a bit of a delicate situation because we don't want to turn people down, but if they need a hospital bed, we never have beds, and I've heard one center, their administration said, okay, you can't give anymore, but I know community center is doing it. So talk to your local physician. And there's a good part of that. If the hematologists get used to giving it, then we get more comfortable with how to do it. So it's a win-win situation. And we are seeing people definitely getting this through these compassionate or access programs. We also just started yesterday, and I'm showing this just to give you sort of a feel for an experience of getting a bispecific. We finally started a pilot of giving outpatient tech teclistamab at Princess Margaret. Now, it's only going to be 15 or 20 patients, 15 patients. We have all sorts of strict procedures. The first 10 will be treated at Princess Margaret. We have our outpatient day hospital for outpatient transplants. And half of them, and this is a trend, we're going to give them TOSI before they get their bite. There is some, it's such a good antidote. And there are a couple of trials that show you can reduce CRS even more than with other pre-meds and step-up dosing by giving a dose of TOSI before you give the drug. So we're going to try that. We've had clinical trials of that before with bispecifics, and we love it. And I think patients may like it because then maybe they don't have that fever that's unpleasant. Then after those 10 patients, the next 5 will be treated at a community center north of Toronto, or north of the city proper, because we're trying to pilot this so we can roll this out when these drugs are funded and available to hematologists everywhere, that we have a way to get it to everyone when they need it, rather than wait for beds to be available like it is now, because there's a bottleneck there. So if you look at the dosing for tech, we're giving it subcutaneously, that's the SC, and it's going to be dosing on Monday, Wednesday, Friday, the step-up dose. So there's a small dose on Monday, see if there's CRS, let it subside. If it hasn't subsided, you delay treatment. Then Wednesday they get a bigger dose, and then Friday they get their first full dose, and then they get it weekly. So this is based on preliminary experience with giving the drug in hospital and with other programs. So we're quite excited and hope this can be exported so that we can make this available for broad usage. So we're excited. Next slide, please. Now, this is a field that's exploding. I've listed here the BiTEs that are under development that target BCMA. I've already mentioned TEC and ELRA. I won't try to say the next one. There's alnuctamab, there's F18212, and there's AbbVie drug. I will say we're very excited. We will have a trial through CMRG for the AbbVie drug that will focus on infection prevention, CRS prevention, and a shortened duration of treatment because these are given until progression now, and we expect that to open later this year. Then there are BiTEs that target other markers. There are two that recognize the purple guy, GPRC5D on the myeloma cells. Talquetamab is under review by Health Canada. It, in a nutshell, has maybe, well, a bit less infection risk. It does cross-react with tissues like the skin, the lining of the oral cavity, and the fingernail. So we have to be mindful of rashes, nail changes, sometimes appetite loss, or some changes in taste. Not severe, but it is an on-target, off-tumor response. And we've had for several years access. I think this was the first one we had that targets the green guy, FCRH5. And I have people that have been treated with this. It was always given a year only, and I have people that have stayed in remission a very long time. So these are incredibly exciting drugs. Unresolved issues, and this will be something we'll all, I think, want to advocate for as we have more data. I mean, in my wish list, I would want my patient to have access to a BCMA drug. When that quits working, GPRC5D drug, and if that quits working, access to Cevostamab. We have, through clinical trials, had patients go through multiple bispecifics. And there is early data that having a bispecific after CAR T, when the CAR T quits working, might be a very good strategy. The challenge will be getting all the data and paying for it. But the science behind it is absolutely amazing. Next slide, please. So I know this is a busy slide, but we do have trials in Canada, and I've tried to show the pros and cons of these clinical trials of bispecifics. You know, trials provide some access, but the eligibility criteria can be limiting. Platelets have to be X, kidney tests have to be Y, and there are no exceptions for going on a study. Once on a study, you can get a waiver if it's in your patient's best interest. To go on a study, eligibility criteria are non-negotiable. And then for these big international trials, may only be 2 centers in Canada that get them. And there's no way for all 20 big centers to get access, to get on the trial. And then each site usually only gets 3 to 5 spots. And the recent trials, now that the bispecifics are showing activity as single agents by themselves in very advanced disease, they need to follow up that kind of trial that doesn't have a comparator, but looks like it extends, well, it does control the disease and it looks like it can extend life. What's called a confirmatory studies have to be done, where you flip a coin, old treatment versus the BiTE treatment. Then if someone goes on that trial, it's only a 50-50 chance of getting the new drug. The other dynamic is, again, now that the bispecifics have shown activity later in the disease course, back to Suzanne's question, what if we give them earlier? And so the focus, although there are trials in people who've had 3, 4, 5 treatments, I've just listed for your interest the trials that are being done now in the settings, like the Magnetism 6, which is open, is elranatamab with RD. Sorry, I put a versus. So Elra RD versus Dara RD for newly diagnosed transplant ineligible patients. We've been given Dara RD very happily to people who aren't going to go to transplant. What if we can give a BiTE up front with RevDex? So then everyone could get it that's over 65 or 70. There's the Magnetism 7, which is after a stem cell transplant, patients are randomized to maintenance with either good old lenalidomide maintenance, which we've used, I think it's been funded since 2013, or the BiTE. So again, if every transplant patient got this, it would move it way up front. Magnetism 4 is studying ELRA with other agents, very limited numbers. Talquetamab is the one that's under review by Health Canada. There's a trial of talquetamab plus pomalidomide versus either elotuzumab, which isn't marketed here, although we've used it, Pomdex, or Velcade-dex in 1 to 4 lines. And so on and so on. And then the last trial is, I think, of interest. It's combining 2 BiTEs, teclistamab and talquetamab, in patients with myeloma outside the bone marrow, which can be quite stubborn. So, there are trials, and we keep trying to get as many as we can. We'll have our CMRG trial, which is in more advanced patients. But we're anxiously awaiting till these are funded, and that we can throw the doors open so that everyone can get them when they need them after 3 lines of treatment. And I think there'll be decisions to be made when CAR T is approved for this setting. Who's going to get CAR T and who's going to get a bispecific? How that will bring up resource issues. If we can do the bispecifics as an outpatient, and CAR T takes 5+ weeks to process CAR T when bispecifics are off the shelf, how are we going to do this? So stay tuned. Next slide, please.
Thank you so much, Dr. Reese. Anybody that knows me knows that I can't work a computer, and somehow I've lost my camera, but I don't want to fiddle around with it very much. Stop laughing at me, Dr. Reese. I'm quite convinced that my computer actually is haunted. But thank you. That was absolutely fascinating. As you know, I've been away from clinic now for a year, and just the things that have come up in the last year with all the clinical trials and all the things that myeloma patients will have access to in the not so distant future, because of all the hard work that you and your colleagues are doing, is so exciting. As you know, Dr. Reese, 45 years ago, I was a very, very young and perky, I must say, oncology nurse. And, anybody that had any type of cancer just went through so much distress having treatment, and usually had very poor outcomes. And the fact that things have come so far in the last 40 years for all cancer sites, and now what I've seen in the last 10 years for patients with myeloma, multiple myeloma. I must say, I'm so grateful to still be able to hang out with the people at CMRG and have some involvement. And so does anybody, first of all, before we go for questions, I just want to clarify, our next seminar is not on the 28th of June. I'm sorry, that was, I didn't realize the 28th is a Friday. So it'll be on the 27th of June. And again, that's when we'll be talking about symptom management, what palliative care is, what it means, and have a little discussion about medical assistance in dying. So are there any questions? You can either just jump in or put your hand up, or I can watch the chat if anybody wants to go into the chat. That I can work. My camera, I cannot.
I can't do chat, so I'm not laughing at you. I'm laughing with you.
I know, I know. Are there any questions out there?
No stupid questions.
No, not at all.
This
Can
is pretty.
anybody come to my house and fix my computer?
I wouldn't touch it with a 10.
So Suzanne,
Yes.
this is Engin. There is a question directed to me as the host, and I'm just going to read that out if that's okay.
Of course.
So the question reads, how many distinct target markers are there generally on an MM cell? Example, CD38, etc. How many distinct target markers?
Well, there are lots of CD38. It's not just one stuck up that the antibody has to find. CD38's in a number of places on the more or less spherical cell. And then there's a whole host of markers. I actually had a picture. You know, you look at a tumor cell and it looks like a hairy ball with all those little hairs are different markers. But the catch is you have to find markers that are only on the cell you want to kill. And there are certain markers like HLA antigens. You know, we do HLA matching, you know, for platelet transfusion sometimes, or organ. There are lots of surface markers. Some of them are necessary for just metabolism. You don't want to make your antibody against those or you'll cause harm. So the challenge has been to find what's called specific antibodies, specific antigens are surface markers, but our cells are coated with hundreds of these that are, some are like receptors, they're like a keyhole, and different little substances come and fit in the keyhole, turn the key and tell the cell to stop growing, tell it to grow more, tell it to divide, you know. So the complexity of each cell and what it responds to through its surface markers is astronomical. I don't know if that answers your question, but now we finally found markers only on myeloma cells, and CD38, finding that changed the world of myeloma treatment.
Thank you, Dr. Reese.
Okay. There's another question in the chat.
Yeah, I'm sorry, again, I'm not actually able to see the chat, so I apologize. Go ahead.
You're just saying that.
So the question is from Tracy, who is asking if this session will be posted later on at a later time. You can answer that, Suzanne.
Yes, it'll be this webinar, as all the others, the seminars will be posted on the CMRG website. I know we did try an earlier time a couple of months ago, but it seems like for most people this is a better time. So certainly feel free to check out the CMRG website that has all of our seminars are posted. And while I've got you all, please remember to do the make sure you let us know what your questions might be, and do the, I've lost the word in my head, the satisfaction, the questionnaire at the end, if you could. Again, Dr. Reese and I and the rest of the CMRG staff really, really, really want to make these seminars what you all want to hear about.
Okay. There's a few more questions, so I'll just read them out for you, Suzanne. Susan is asking, all of these treatments seem to involve a weekly hospital visit. Is there anything under development that has a monthly schedule?
Yeah, I think there is a major push that after the step up, where drugs are given every couple of days till you work up to the full dose, they're given every 2 to 3 to 4 weeks. And I believe they're being studied. I'm trying to remember, showing data that whether you give it every 2 or every 4 weeks, it may be sufficient. So the effort is to try to get these once a month, not every week. I think that's just for long-term use. And the other thing I didn't talk about is, I'm happy to say this, although it's not tested. I have always been such a proponent of continuous treatment. I'm very afraid to stop treatment because I don't know what's going to happen and whether my next treatment will work. And the data that we've had, although it's not your nice, good randomized trial, has generally supported continuous treatment, particularly with Revlimid, pomalidomide, and CD38 antibodies. But the bispecifics work so well, maybe too well, and there's a wave of studies coming out, including our CMRG AbbVie BiTE trial that Guido Lankman's going to lead, that will not give it until progression, because there may be a sweet spot that you don't want to completely get rid of your immune system and make no antibodies. And maybe the better strategy is to give it a certain time. These can produce, particularly if very deep remissions, MRD negative, are produced, maybe you need to take a year break and start them again. Maybe it's too much of a good thing. Again, this is all ripe for future study, but it's the first time, this is my opinion, that I actually support, maybe not giving it forever because they're so effective. They're not curative. But the other trend that we're going to see, and why this area is so ripe for study, is you may have noticed on that last slide, people are giving these drugs with POM, with lenalidomide, with each other. And that it's very hard to, that preliminarily, if you look at grouped analyses, that seems to have higher response rates, longer remissions, again, very preliminary. But then, you have to balance that against whenever you add drugs, there's more toxicity. And then you have to add, if you're giving it with daratumumab, what's the cost per month? So, the challenges, it'll get worked out, but I think still the main goal is suppressing the disease with a good quality of life as long as possible. And, we've already made so much progress that we can see in our database over the years. And, it's just such an incredible time for sorting these things out. And we'll talk about CAR T later and where that will be. And, that will become a reality. It looks like in Canada. And we have a whole new doorway open to immunotherapy, and who knows what our treatment algorithm will look like in 5 years.
Thanks, Dr. Reese. And you have the next question?
Yes, the next one is a comment. Thank you from Sonia. Thank you for your amazing presentation. It gives me so much hope for my future as a myeloma patient. And there's a question from Ellen. What is the advantage of CAR T versus BiTEs, other than the fact that CAR T has a long lead-up time? Suzanne or Donna?
What
Dr.
is meant by a lead-up time?
So,
Well,
I'm assuming the fact that CAR T is off the shelf, and then BiTEs are off the shelf, and
I
CAR
see.
T you have to have about 3 or 4 weeks to
Got it.
prepare, 4 or 5 weeks to prepare.
Yeah, I mean, people make columns comparing the pros and cons of each approach. And right now, if you look at sort of the group of people that get who've had 6 lines of treatment, and you just look broadly at Cartitude, not ide-cel, but Cartitude, that's the one that gives almost 3 years PFS. And the BiTEs are more like a year and a half as one drug. Now, everything's still moving. I think what we want to see is, but people, not everyone can wait 5, 6, 7 weeks to get a CAR T made. And, there's a practicality issue. And I think what we're hoping as hematologists is it won't be all or none. I mean, that people can get both at different time points. How that will play out is unknown. And I think we're just going to have to keep knocking at the door of our funders. People can't have just one BiTE, when you have a different target, and we haven't even talked about mezigdomide, which is like the super, super, super duper pomalidomide, which is an amazing drug. So there's, I think we just don't know. So also the bispecifics are not going to stay single agents. And it's not clear that CAR T will be a single agent, so I don't think we have the answers, but for a given patient, there will at some point probably need to be a decision based on the funding, at the time that it's needed. Will a CAR T be feasible and more appropriate, or will a BiTE by itself or in combination be better for that individual patient? That didn't answer.
I know, I think you answered it very well, Dr. Reese. And I think the thing to really take from all that is that, there's so much excitement in the myeloma world with all of these things going on. And as the last patient or attendee said, I mean, all of this is giving such hope to patients going forward from here. And again, the last 10 years, especially when daratumumab came around the corner, it really did change everything. So time will tell. All I can say is to count on your team. Be as educated and informed as you can be, and CMRG can help you with that, to know what is potentially available to you and or your loved ones. So I think you answered that very, very well. Engin, are there any other questions?
So there are 3 more questions. I'll go through them quickly.
No worries.
So the next question is from Robin. And the question is, are there any indications why certain patients respond to one BiTE rather than another?
Ask us something easy.
Yeah. I might as well be answering that.
I've seen that. One flat out does nothing and the other works like a charm. I don't think we know. Things that have been proposed but not proven is that for some reason the myeloma cell is mutated and isn't making that marker on the surface anymore. Marker loss, antigen loss, that is not felt to be the most common, although that makes the most sense to me. You don't have the marker, the BiTE can't link the cancer cell to the T cell, and the CAR T won't work. But I don't think we know all that. There's a lot of interest in T cell exhaustion, that if the T cells that you bring to the myeloma cells are worn out from all their treatment beforehand. That's a hot area. And, there's a lot of excitement that maybe things like IMiDs we know they help the immune system. They help the T cells. Can they regenerate the tired out T cells? So there's a tremendous amount of science that we need to sort this out. And we're gonna, I'm very excited, as you can tell, about our CMRG trial. We're going to be able to do some correlative studies that look at T cell subsets and function. And you can't look at everything with every study, but try to get some science correlative. So we're doing more than just treating and saying, X number of people responded, some didn't, but we don't know how to interpret it. So anyway.
The work goes on. The work goes on. And what was the next question, Megan?
So the next question is from Vito. Why are there just naked antibodies and no BiTEs for CD38 markers?
That's a good question. They have looked at, I believe, some CAR Ts that target CD38. I don't know how much of that decision is because the naked antibodies are so well entrenched. They'd have to compete with, something that's so well established. Some of these developments by necessity have to account for marketing. Are people going to, I that's an excellent question. I think I don't know how much might have to do with if you make a BiTE to CD38, it doesn't have the same killing power. I've heard nothing in that regard. But I don't know how well it's been studied. And it may be as simple as the CD38 plant antibodies are integrated into every phase of therapy now. And there just isn't a niche to do all the research and then sell enough drug to make it worthwhile. I don't know.
Thank you, Dr. Reese. And next, Engen?
John is asking, could you repeat the opportunity you mentioned about clinical trial for post-transplant patients? Is there an anticipated benefit of BiTE over DARA?
Yeah, good question, because we talked before about another unmet need, which is for transplant eligible patients to have DARA added with induction and then with maintenance. So that seems to have been the closest thing to a home run we will have with transplant eligible. So the question is, if we put a BiTE in there after transplant rather than DARA, with or without Revlimid, would that knock the ball even further out of the ballpark? That is obviously on someone's mind. We have the trial that some of it has to do with when the trials are designed. And then from the time the idea is there till the trial's finished is several years. So I think when the trials now of using BiTEs either as maintenance by themselves or combined with Revlimid in the randomized trials, and there's another one coming with teclistamab as part of maintenance. DARA wasn't being used with transplants when those trials were designed, before and after. So the next generation of trials might be after transplant, randomizing people to Revlimid alone, Revlimid and Dara, Revlimid and a BiTE alone. That's how you'd answer that question. But that question was not relevant or ready to be asked at the time the current trials were written. I think, and then, you know, how complicated it is waiting for these randomized trials, like in newly diagnosed patients. If transplants are having people stay in remission 5, 6, 7 years, you know, by the time you know the results, the science has changed so much. You may be aware that the FDA has finally said, okay, for trials, we'll accept MRD. So as a surrogate, we don't have to wait for these trials early in the course of myeloma, 8 years to see the result. We'll have a surrogate. Now, confirmation will have to be done later, but that gives us an endpoint that will hopefully let us escalate the field faster. And we're trying hard to get this into Canada. It's not easy. Not to determine treatment, but to let us do trials faster.
Thank you, Dr. Reese. I'm just noting the time. We're almost at 7:30. Are there any other questions?
So one last question.
Yes.
Two-part question from Helene. Looks like we have lots of horses to pick from, but is there ongoing research studying patient blood work that would determine which horse is the best? And is myeloma the only blood cancer using BiTEs?
Wow.
Well, I'll do the first one. The second question is easier. BiTEs are being used in acute lymphoblastic leukemia, ALL, and in lymphoma. They're finding a way. Picking the right horse to bet on, no, there's no way. And with the single agents, because I think I didn't show the slide. If you line up the single agents in people who have had 6 lines of treatment from all the reports where they're studied by themselves, they all give a response rate around 60%. And they all work a year, 15, 16 months. So you can't by that say, I really want my patient to have this. And that's one reason we've wanted patients to be able right now to have one, and then when it quits working, go to another target. When that, if we only have one, we need, that's called biomarkers. We need a biomarker to tell us, is the BCMA target the best? Is it on the myeloma cell? Is it on there in the right concentration? There's interest in looking at T cell health, but we are not there yet. Just like we're not with regular treatment. How do we know who's going to respond to carfilzomib and who's going to respond to POM? I mean, we end up trying them all.
Absolutely. Well, there are still a few people left online. I want to thank everybody that participated tonight, everybody that was online. Thank you, of course, to Dr. Reese and to Egan, who hosted our webinar or seminar this evening. Again,